Welcome. Good to have you. Good to see you.
They get all of that stuff in Internet Crypto. They get all those things a lot more.
Opening ideas.
Well, yeah, because it started as
Yeah.
but you know, they're investors. They follow.
Yes.
They follow how to get returns. And like in 2021, that was probably crypto, and like now it's AI. Fine. That's their thing. So
Pivot.
yeah, well, imagine they didn't pivot. They're still being like, oh, we're crypto.
Yeah,
They
that's-
maybe like,
Well,
these people
now
know.
it's
I know it's
back.
back. It's back. It's
We
back.
try to
Actually, even our
verify
bank
that the
was.
structure is what they say
It's
it is.
gonna be a
And because in this case it's spectrogram, you have hypercodes versus hypercodes.
You can see like over what
You can see a visual. And so that's also very well.
yeah, it's really classic. Yeah, very important.
Yeah.
Yeah, so just similar visualization.
Yeah.
Code generation visualization.
So when you have models interpret, do you mean just existing off the shelf generative models or testing?
Yeah, no, it's custom training. You just take the graph that comes out of the cloud.
Gotcha.
For an interpretation.
No. Like,
It's just very different.
Did it come up with any insights that you wouldn't have seen yourself?
Oh, well, kind of, because it's more
Right.
yeah,
Sure. But I think what I mean more by that is,
and there were insights.
that's the question.
Yeah.
That's it.
But I think in part it's because of the
Right, because all else do is match a lot of patterns which you don't have.
Five simple patterns overrides all. Bring it moving. Yeah, to take a seat. Hang out. I'll call folk.
Welcome to the AI for Medical Science and Longevity Hackathon. Great to have you. So if everyone can actually come to this sort of front area, sit down, get cozy, make friends with your partners. We have a number of fascinating nascent projects. Just discussing with one of you the use of image modeling in order to understand structure data over DNA. There's this massive overhang of AI technology that can be applied in simple ways to biological research problems. And so I hope we can knock a huge number of them out as a collective today. We're going to talk from the folk at Chai Discovery, who have put out a nice custom model for biodata. And it is in part my hope that if you have a great idea for fine-tuning or for training a novel biological foundation model, that you take a shot at it. A big part of why we wanted more time in order to code today was so that we would have time to train models. And so do let me know if it's of interest. But just to sort of set the stage with some context, this is AGI House. We have this incredible series, long series of productive hackathons, and in this case, AI and longevity, we are focused on all of the ways that bioresearch has already been impacted by machine learning. So an example is OpenCRISPR-1. You could take a large data source of proteins, train a model in order to generate proteins that are in that style, and use it in order to explore a very large space. And there, in general, is this emerging paradigm in ML-powered research, where if you can automate the evaluation process, for example, checking whether a proof was correct or not, or checking whether your program has sort of fulfilled an interesting outcome, then you can do search with an LLM that is either customized to your task or that is off the shelf, that is appropriately prompted, and then auto-evaluate tons of potential solutions to an interesting problem. And so do feel free to innovate within that paradigm. It's been really fruitful, and it might be easy to knock out a novel project where you say, find a way to evaluate the quality of the protocol and do protocol generation for tasks of interest, and on the basis of your evaluation quality, discover high-quality new protocols for tasks that already exist. There are a number of models that are of interest, so the docking problem would be a fantastic place to innovate with either fine-tuning or monitoring. Ego was trained by ARC, and I got dinner with her this week, and in a lot of ways, it's an example where machine learning for us can make a big contribution to biology without necessarily having tremendous amounts of background. And so the hope is that those can mix with one another. Some of you are basically pure engineering, some of you have a clear angle on a novel biological foundation model, and train something special. So that said, we have a few folk over from Chai Discovery. They are newly formed, and the founders of the organization coming out of the MetaBio team, out of a really fantastic history where some of their founders were behind the architecture that was used in AlphaFold, among other things. And so we have Paul and Joshua here with us. And I will let them say hi, introduce themselves, and give us a little bit of insight into what we should go today. Let's hear it for them.
Sorry we can't be there with you in person today. Just want to do a quick mic check. Can you guys hear us well?
Yes.
I think so.
Is that a yes?
Yes.
Turn off your mic in your pocket.
Oh, it's this one?
Is that a yes now?
Yeah, we can definitely hear you, but there's a bit of an echo. So
Okay.
we're getting rid of
I hear some feedback from myself as well.
It's this link. Can you try speaking again?
Yeah. How's this? Is that better?
Oh,
I'll hear the feedback.
So just mute your mic.
well, then, so now he can't hear us.
Okay. Is that good? You can give me the thumbs up, thumbs down.
Okay, that's
Okay. Awesome. Cool. Well, I guess if there's questions, you'll just have to let me know. So maybe just motion Jeremy, and then we can figure it out. Okay, cool. Well, let's start with some quick introductions from myself and Paul. So I'm Josh Meyer, one of the co-founders at Chai Discovery. I've been excited about AI and biology since I was a kid. Grew up as a programmer, went to a high school with a stem cell lab, so got hooked on molecular biology very early on. I spent some time at the Broad Institute working on CRISPR. I was doing both like wet lab method development there, and we also trained some of the first computational methods of actually designing CRISPR experiments. That's where it kind of hit me that these weren't separate interests anymore, AI and biology, but they would come together very soon. So from there, I went to OpenAI. I was there in the nonprofit days. We worked on GPT-1 while I was there. So I went there at first because OpenAI was like the best in reinforcement learning, and we started doing language models, and I realized that if this was working for natural language processing, it should probably work for the real natural language, which I think is DNA and proteins. So I ended up leaving and going over to Facebook, Meta, to help establish a protein folding division. And over there, we trained the first transformer LLMs for protein sequences. It was I think a pretty exciting time just to see these methods like working for the first time. Very exploratory. We didn't really know what the methods would be capable of at the time, but spent a couple of years doing some of the basic research around the models. And then after that, I was recruited over to this company called Absci. It was an antibody cell line development company. So again, very much like wet lab company that was excited about what AI was going to do for the space. So I ended up joining them, was chief AI officer over there, and we have a pipeline of drugs that we created with the help of AI. And now going into 2024, we came together. We started Chai Discovery. We're a small group of folks. Many of us have actually been in similar kind of roles, running AI at different drug discovery companies. And we are trying to build foundation models for biology that can really enable the entire ecosystem. So if you look at the mission, our mission is to turn biology from science into engineering. These days, if you count, there's probably like 100 companies that are doing AI for biology. We think in 5 years from now, every drug discovery company will be an AI drug discovery company, and that's because they'll be building on these kinds of technologies. But it's also becoming harder and harder to develop those technologies over time. So that's one of the reasons why we put out some of our work open source. You know, we'll tell you about Chai-1 today, which is the first foundation model that we put out. I'll note that we're not just a protein folding company. You know, we put out Chai-1 for free. It's open source. We really want to enable the ecosystem here. We built it because it's uniquely enabling to some of the stuff that's coming next at Chai. But once we got these results, we decided to to make it available to the community. So excited to walk you through it. Guess game plan here is we've got about 30 minutes, I believe, and we'll take you through some of the results from the model so you can see what it's capable of. We'll give you like a demo of actually doing like protein folding, and then I'll also walk you through our code base so that if you're interested in using it for the hackathon, you'll have some pointers of where to go. So that's a quick intro about myself. Paul, you want to introduce yourself quickly as well?
Yeah, hi, I'm Paul. I'm very excited to be here, have this opportunity. Thank you to AGI House. And I've been doing some of the very early work in small molecule generation and small molecule optimization for molecules to be, for instance, synthesizable or look like drugs. And for that, I applied diffusion models and classifier guidance. And then I've been going to CERN. There I was part of the foundation models team, and there we trained some technically very similar models where we have point clouds and diffusion models to conditional generation of these point clouds, which are not molecules but outcomes of particle collisions, but essentially very similar work. And now I will become an AI researcher at Chai, and I'm very excited for that.
Cool. Well, let's get started with the presentation. So I'm going to pull up here the technical report first of all. So you can find this on bioRxiv. There's actually a slightly different version with some results that are hot off the press. We're still pending for this to get onto the preprint server, but basically broadly same concept. So Chai-1, it's a multimodal foundation model for structure prediction. That means that it doesn't just predict proteins, but it can predict the structure of proteins with small molecules, DNA, RNA, post-translational modifications. I'll show you how to use it soon, but let's walk through what the architecture of this model actually is and what we're trying to do here. So here's an overview of the model architecture and input features. There's a ton of optional data that you can put into the model. So you'll see this when we walk through the code base, but there's many ways that you can use the model. In the simplest form, you would just put through a sequence of a protein, for example, and then you'd get a protein structure that comes out of it, along with some information about confidence, how confident the model is in its error. We actually train the model to predict its error. This is quite common in biology these days because if you use ChatGPT and you get some text out of it, you can tell if it's like if the syntax is correct, if the answer sounds correct. Very hard to do that in biology. If I just show you like a protein sequence or a 3D protein structure, very hard to know if the thing is actually predicted correctly. And that's why we actually have the model predict its own errors. So during training, the model is making predictions of these structures. We obviously can measure the error because we know the ground truth, and we're training the model to also predict its error for those structures. So there's a number of other inputs that you can give to the model to improve its performance. You can give it embeddings from a language model. So this is a protein language model. You take that protein sequence, run it through the language model, and this improves performance. So basically, in any case, if you're going to just put in a single sequence, you just want to do things quickly, you should probably use the language model. There's really no reason why you shouldn't. Maybe it would be like slightly slower, but it's actually a very small part of the processing time compared to the total algorithm. You can put in structural templates. So these are similar structures to what you're producing. We actually, so we train the model with structural templates. We actually recommend that people use functional and structural prompts, which I'll get to in a moment. These actually are easier to use, I'd say, than the structural templates, but you can think of this as like RAG, retrieval augmented generation, where when you're trying to make a prediction, you go and you look at similar stuff in your database that might help you make a more accurate prediction. So we can do that both for similar structures, and we can also do that for similar sequences. So you can take a sequence, you can find a group of related sequences, and then provide that to the model as well. That's called a multiple sequence alignment. And about a week or two ago, we added support for an MSA server into the Chai Lab repository. So you'll see that soon. If you want to go and do these kinds of lookups, you either go and download a couple terabyte databases and then run some tools against them, or there's a server where you can go and you can kind of build that yourself, or you can basically build that automatically. So these are the main inputs to the model. We can also put in additional functional and structural prompts to further boost performance. So these can be, and I'll highlight an example in a moment, but these can be examples of like, here are two residues where I know that the proteins are in contact with one another. If you give that prompt to the model, you end up getting like better performance because you've kind of restricted the search space in a sense. And we're not, explicitly constraining the search space, but it is additional information that the model can use. So that would be, that's what we call here cross-linking from mass spec. There are experiments in the lab that are quite cheap to do where you can get that information. I don't think anyone's going to be able to do those experiments today. I don't see any lab space around, but I'm sure you can find some stuff in the literature, for example, if you wanted to play around with that, or you could also just use some of your own knowledge to kind of choose those. Similar to that, we also have what's called epitope mapping. So in the tool, we call this pocket conditioning. So this is, let's say we have two proteins or a protein small molecule that are interacting. You can prompt the model and say, on one of the protein chains, this is the pocket, like this is where an interaction is going to happen. So instead of it being, an interaction on two partners, you can just, kind of give the information about one of the partners. You can put in what's called apo structure. So this could be the structure, like basically the full structure of one of those chains if you happen to know it. This is called apo without getting too much into the details. This means that we know the structure in isolation. If you have like multiple proteins together, the structures of each of them might change a bit, but maybe you've got some information about what one of them looks like, and you can give that to the model as well. So a couple other things that, interest of time, we can kind of skip over, but a bunch of various like functional and structural prompts that the model was trained with. And then lastly, there's information about small molecules and modified residues. So if you want to provide a small molecule to the model, it's a little bit more complicated to specify that. If you're talking about a protein, you've got 20 amino acids, and you can just write out that sequence. It's kind of like text. It's very easy to think about. For a small molecule, there's various ways in which people denote small molecules. One of the most common ones is SMILE string format. So you'll see these examples later when we show you how to use the model, but SMILE strings are just like one kind of way of explaining what a small molecule looks like. The way that the model actually reads this, it's not literally like reading that SMILE string. We actually transform that into conformers. So a conformer is it's basically some calculation of like what the small molecule is going to look like. There's a tool called RDKit that we use for kind of computing these, and then we provide that computed information directly to the model. So I'm guessing today, if you guys want to do small molecule prediction, you would just put in a SMILE string. But if you wanted to hack in support to put in like some custom conformers, then you could do that as well, and it should be pretty easy to find where that happens in the code base. So those are all the inputs. Is there a question?
Yeah. I wanted to open the floor for initial questions. If anyone's there.
Any chance you could give some examples for language models? What kind of sequences you're talking about?
So he's looking for examples of language model sequences. What kind of sequences?
Inputs.
Just like amino acids, A, N.
Yeah, so these are all protein sequences. So a protein sequence is a string of amino acids. Every amino acid is like, there's like 20 possible. Let me just, why don't we just jump into the demo actually? Let me just go into that, and then we can come back to architecture and stuff. Maybe that will give some more color.
And then I also had a question. How much of sort of conflation is there between the small molecule input data and the protein input data? I guess amino acid sequences feel quite different to me from cells. I wonder if you get any issues in combining those into a single model. And I don't know how distinct they feel from you to content that typically go into a general language model or multimodal model even of images. If you expect there to be emerging of foundation models where all inputs are possible and the model takes care of the distinctions between them, or do you end up believing in a small molecule specific foundation model, or like what trajectory in the sort of space of where you think state of the art will end up exists right now?
Yeah, good question. So I think the demo is probably going to answer both of those questions. So if you look at, I assume everyone can see the demo now. So this is a web interface where you can use the model. Again, this just like wraps the GitHub repository. But if you, so let's take an example over here, just so we have some stuff pre-populated. You can see here's like we actually specify the molecule type to the model. So this is a ligand, for example, a small molecule, and then these are protein sequences. So the vocabulary is different for both of these. So again, protein sequence is it's a string of, everything here is like strings, but this is a string of like 20 possible amino acids. Smile strings are a little bit more complicated. So for example, you can see this is basically saying an oxygen is like double bonded to like one of the carbons over here. See if I switch to this other example, you can see here's like a more complicated kind of smile string. What you could also do is put in what are called CCD codes. So if we go to, this is, the name of this protein is 7qF4. We've actually just copied in here the sequence of a protein that's already been determined. We can go to the RCSB. This is basically a big data bank of all these proteins. This is actually, if you can see here, in 2022, someone actually determined the 3D structure of this protein. That's something that can cost tens of thousands of dollars. It can take a long time to do, but people are doing these experiments all the time. There's hundreds of thousands of structures. Actually, you can see here, 228,000 structures in the protein data bank. And this is something that's outside of the training set. That's why we use it as an example on the server. But you can see the small molecule, for example, this guy over here. This is the one that we put in, riboflavin. You could also put in a CCD code. So if you put it in, you can see this thing's called RBF. And RBF refers to this. You can see this is the conformer. So, you know, we're writing it down here. If anyone's taking orgo, like this is what it might look like if you write it down on paper. And then this is sort of what the 3D structure looks like. And this is very easy to compute. You don't need any fancy AI to figure out the 3D structure. This is just like, you know, good old chemistry. And then you can see the CCD code is called RBF. And that's just like an encoding of this thing that our models are aware about too. So you can either put in like the full encoding this way, or you can just do like RBF and that should work too. So that's a bit about, yeah, does that answer the questions about like what, oh, and I guess like how does the model like process both of these? So we put them in separate tracks. So there's different molecule types here, protein, ligand, you can actually put it in DNA, RNA, and for each of these, you know, the model is going to, in each case, the model will process them independently. So there's basically a different kind of vocabulary that's used. Like take the small molecules, for example, there's no embedding for every token over here. The model will take this, it will convert it into some conformer. You have that reference structure, and then it does some processing on top of it. For a protein sequence, we pass this through a language model. You get embeddings for every amino acid, and then you process that through the model. If you turn on multiple sequence alignments, then we do that genetic search, so we find a group of related sequences. Now we've got a whole bunch of protein sequences lined up, and we basically embed that entire matrix. So you can end up with actually many tokens in input as a result. So this is the way we deal with multimodal data. You know, every time you add a modality, you have a different way of processing it, and it all gets integrated at some point. But on the input embedding layer, we call it, these are processed differently. So it's very similar to multimodal models for like text and images, for example. You'll process the text and the images separately, and then all kind of combine together at some point.
Can I ask a question? Hey Joshua, this is JJ. I have a question about like, cause you know, like the amino acids and nucleotides, etc. They're kind of composed from like atoms. Like, do you think the cross-grain representation may somehow improve by considering like the atom embedding, etc.?
Jeremy, do you mind repeating the question? I didn't hear it super clearly on the Zoom.
Yeah, I think that because these are all, at the end of the day, composed of atoms, that having an atomic level would you know, be preferable, would be more general, than operating at each level of structure.
Yeah, so the model does operate at the all atom level. So this is just a way of specifying information. Again, if we go back to, you know, this RBF over here, the reference conformer, like this is a full atom representation. Amino acids are also converted into full atom representations. So it's just like to make it more convenient for specifying things. Like, again, if you want to specify these amino acids, you don't know how they all relate to one another. You know what, I can show you an amino acid chart. the model now is converting, if we have like an R, that's arginine. It's converted into this implicitly in the model. So the model, kind of starts with that. It's a diffusion model. So when we're training, for example, we know what the full 3D structure is like. We have all the atoms. We run some forward diffusion. The model has to go and like predict what the thing looks like. But when the model is conditioned on all this information. So it knows what the atoms are for these things. It just doesn't know how they relate to one another. Because I can just give you a list of like arginine, lysine, aspartic acid, right? And we know what these chemical structures are like, but we don't know how it all comes together as like one big protein. And that's kind of the point of the machine learning model. So it is actually operating on a full atom level. In the code base, we'll get to that very soon. You could condition on all atom information as well. So you could say, here are how like the distances between various amino acids. That's we have here a demo for restraints. And that's like specified here, for example. You could say chain 1, A, position 387, B, position 101. These are 5 angstroms away from one another. An angstrom is the diameter of an atom. So you could provide that kind of information as well to the model. And that's sort of like a full atom prompt as well.
Gotcha. So, hi, I'm Roman. I got a follow-up question on that. And so when we look at both atoms and molecules, there's a bunch of explicit features we can notice, like how many free electrons are there, oxidation state, stuff like that. And based on what you're describing, it sounds like this model kind of gets those as implicit features due to training on a bunch of examples. Do you guys have any kind of infrastructure in place to define explicit features as well?
Yeah, so again, those features are determined automatically from the inputs. Actually, things like charge state, like that is actually provided to the model. And again, if you could like read that off of this, for example. So these are all things that can be determined ahead of time. My guess, and I'm just speculating here, my guess would be like, we probably didn't have to train the model with that because it might have just been able to kind of learn it automatically from the data. But we provide it anyways. And this way you might be able to do some fancy things on the input. For example, we had a lab that's like, I want to put in a partial state. Like some of the electrons are distributed between these like 2 atoms. Can I specify that to the model? And the answer is like, we haven't seen those examples during training, but like, try it out. Let's see if the model can like, figure out how to deal with it. And we wouldn't be surprised if it does. So yeah, that information is like provided as well.
Awesome.
Let's keep moving.
Well, maybe I'll just go through results really quickly, and then we'll go back and show some demo about how you can actually use the model. So we talked about a bit of the input features. Architecture, I'll just kind of gloss over, but a bunch of different pieces here predicts the structure. We have a complement set that predicts output. And then we evaluate the model on a couple of different tasks. So we look at protein-ligand interactions, we look at protein-protein and protein monomer performance. We're looking here at success rate. So the metric changes depending on what task we're looking at. So we use, for the protein small molecule setting, we're looking at whether the small molecule is placed within 2 angstrom RMSD of like the correct answer. So that might be a mouthful, but we're basically in that case looking at metrics that like chemists are often looking at to say that this prediction is like good enough for doing drug discovery and understanding what's going on. For protein-protein interactions, we use a metric called LDDT. It's kind of a metric that's called local distance difference test. And it's basically some binned calculation about like how far various residues are from one another. So you can see that the model has pretty competitive performance with other state-of-the-art models. So if you look at Chai1 versus AlphaFold 3, they're roughly neck and neck. I think Chai's like 77 versus 76%, but overlapping error bars models basically performing pretty similarly. It's interesting, like a big step up from the previous generation of models, like Rosetta Fold Atom. This is from the Baker lab. And we also find that the model has better performance compared to AlphaFold 2, for example. You can jump over to, we have some plots in the supplement as well. So these will be posted soon, but we compare over here to a recent evaluation set. One thing that we kind of point out in this figure is that it can be, if you just look at some of the benchmarks that people like to use, such as like Cas15, the error bars are actually huge here, and it's very hard to say that like one method is like better than another. What's actually pretty interesting is if you look at this like Cas15 benchmark, there's only like n equals 26 over here, and AlphaFold 2 actually looks like the best from these different methods. So I think it just kind of highlights that it's important to use much larger sets. When we then look at instead, a set where n is like 600, then you can actually see statistical tests pass between these different things. And you can see this is Chai1, this is Chai1 run in single sequence mode. This is AlphaFold 2, and this is Boltz1. So you can see, some separation across the methods. And again, in this setting, what we're looking at here is the metric here is the fraction of cases where we get an acceptable docking performance. Basically, does like the structure kind of look okay according to this metric? And we're finding that over 60-70% of the time it's actually looking good. So this is not going to work all the time, but the nice thing is we have the confidence metrics that can tell you whether things are working. So if we jump back to the demo for a moment, let me just show you some predictions that I've made in the past. If we go in and look at this, you can see the model. So this is an example of like 2 copies of the same protein. And this prediction is actually not done very accurately. And you can see that actually the model knows about that. It knows that like each chain is kind of predicted like decently well. That's why the colors are quite light over here. But when it looks at how it's placing the 2 with respect to one another, it's not very sure. It actually expects that its error is going to be higher. So I actually knew what the ground truth is of this protein, and I went and I compared it to the ground truth, and the prediction wasn't that great. Let me see. I think I actually have it pulled up over here. Let me show you inside this program called PyMol. So PyMol is a software where you can download those results from Chai, and then download this program called PyMol. It lets you kind of visualize this stuff. And I loaded up both like the ground truth is this, and then I put in the prediction. And then I aligned the 2 things. So I did align 1DP4 and the prediction, and you can see the RMSD is quite bad. It's about 20 angstroms RMSD. And you can see the green versus the blue, they're not overlaid that well. What's really interesting though about this is if we then go back to the demo, the model is basically figuring out that like the error is something about like 20 angstroms overall when we're aligning, on one chain versus the other. So it's nice that the model is able to figure this out on its own. Again, that lets us kind of figure out when we can trust the model and when we can't. In the last couple of minutes, maybe we should go through some demos of the code base. So you can find the model, you can either use it on the web server. This is lab.chaidiscovery.com. So I'd recommend starting there just to like get a feel of how you use the model. If you don't want to do some building on top of it, you can go to the Chai lab. We've got a README that walks you through how to install it and how to run the model. There's a command line interface over here. I'll hand it over to Paul in a moment to kind of walk through some of the examples. Paul, do you want to share your screen actually? Or I can just pull it up. But basically there's a command line interface. So you could take a FASTA file. A FASTA is just it's like an input representation that people often use in biology. So if you look at like, let's go to the examples, and then, let's look at, here's like an example of a FASTA file. So you just put in every line, it gets a header. So this is, you can name these with arbitrary names, like whatever you want. So in this case, we're calling it protein 1AC5. We have a protein sequence here. This is a glycan. Actually, I said you can call it whatever you want. The first name is important because like I showed you on the demo, you have to tell the model what track to use. So this tells you use the protein track, and then you can name it whatever you want.
Can you zoom?
This is a glycan.
Zoom in on the code.
Sorry?
Can you zoom in on the code?
Can we zoom in?
Yes, thank you.
So here's like a protein sequence. Here's like, these are glycans. Glycans are like these sugar molecules that you can like put on proteins. There are other examples here as well. If you look at like an MSA, then this is going to be quite a big file. So we have we'll walk you through the examples of how to prepare these in a moment. So at a high level, you can just specify one of those FASTAs, and then you could use the command line interface. So this will be similar to using the web interface, for example. If you want to run with multiple sequence alignments, which you get improved performance, you can just use MSA server. So put -- use MSA server. It will send that information into the interwebs to go and run the server and then return back the results. But there's also the programmatic interface. So Paul, feel free to take over the screen share, or I will.
Yes. Can you see my screen?
Yes, now I can see your screen.
Okay.
You just need to do that. Okay, yes, perfect.
Great. So yeah, let's go to the repository and then to examples. And here we can, this is the simplest example. Here we're running a simple structure prediction. So here, just as Josh showed,
yeah,
we have
please zoom in to all code. Please zoom in aggressively to all code. Thank you.
Yes.
Thank you.
So here we have a FASTA file, just as Josh showed, where we can have ligands, proteins, and we can name them. And it's just split into a file. And yeah, we have some output direction. And yeah, we use this simple function run inference, which we'll see later as well, where we can fix some of the hyperparameters. So the number of trunk recycles. The trunk is basically the language model which encodes the sequence, and the output is fed back into it. And increasing the number of recycles might, in some cases, improve performance. Then we have the diffusion model, the number of time steps. So how often the diffusion model, which is conditioned on the trunk output, is evaluated to go from noise all the way to the structure. If you increase this, you will generally, it will generally take more time, but it might increase in performance. Seed is self-explanatory, since the diffusion model starts from noise, also device, and the ESM embeddings are the language model embeddings of the structure. Yes, our output are CIF files, which are which contain the structure and which can very nicely be visualized, and then some scores of how confident the model is in its predictions. Are there some questions? Otherwise, I would go to the multiple sequence alignment. So here in the multiple sequence alignment,
Yes.
oh yes, then
It's
go
kind
ahead.
of related to what you were talking about. So I was wondering about when you have the docking achieved with this model, is there a way to identify the residues where the two proteins meet? Like, do you have that information put out of the model somewhere? Or is it just kind of a visualization that you can see?
I'm not sure if we have anything set up that does this automatically. Josh, do you want to jump in?
Yeah, do you mind just repeating the question one more time?
Yeah, the question is about residues under docking. So do you just have a visualization of the docking, or can you see exactly what the residues are when the docking process is complete?
I'm not sure I fully understand it. Let me try to answer it. You'll tell me if I'm answering the right question. But the model can be run, when we say the model can be run in docking mode, essentially what that means, so what is docking? Docking is like, I know two structures, for example, and I want to now put them together. So the way that we actually do docking, we just treat it as a folding problem, but we condition it on additional information. So if you give the model those two structures, and then you ask the model to now fold, but it's conditioned on information, it's essentially docking if you do that. So it's the same process, we just condition on additional information. Let me pull up the, I'll take over the screen share, if you don't mind, for a moment. But so let's go back to, if we want to do docking, then we use restraints. So I think this is probably the only folding model right now that's out there that can do this stuff. But we call this Chai1R, R for restraints. And there's a whole README here that explains like how you would do this and some example results. But it's similar to what we showed on the repo, where you could put in either contact or pocket information. The other kind of restraint that you could use is also like a docking restraint as well. So let me see if I can find it quickly in the code base. I guess we don't have an example. Yeah, so if you look at, okay, we have pairwise interactions.
I don't know if we input the information where the because you give it the two sequences, do we already know where the two sequences meet each other for the molecules to contact one another for the docking, and then it just shows us visually that process, or does the model show us visually the process and then we can understand what sequences are touching?
Yeah, so the model is supposed to predict the full 3D structure, right? In which case you can look at the structure and then you can see where the things are interacting. If you would like to condition on additional information, then you can, because in certain drug discovery settings, we know where the two chains are going to interact with one another, and we would like to provide that information to the model. So that's we have this restraint context over here. The examples in the README are pocket and contact. I realize we don't have an example of like docking but in the README, but you can just look at the code here. So there's a docking restraint. If you look at, docking restraint is defined, we have like a docking restraint generator over here. And essentially you can just give this information to the model. If you condition on basically the structure of either of all the chains, then the model can use that information in order to make a better prediction. So this question about like, is the model predicting or is it not predicting? Like, it's always predicting this stuff. It's just like you can tell it the answer, and then that makes the task easier. So if you look at, we actually I think showed this in the README. If you go back to Chilab examples, restraints, then if we just tell the model, even just like contact restraints, not giving it the full docking, but just telling it like, here is like some contacts about like where the things interact with one another. Then you can see a prediction goes from like a score of 0.02 to a score of 0.40. So you really can get much better performance if you give this information to the model. The reason for this is like, again, the model, especially in some of the harder tasks like antibody antigen prediction, the model struggles to predict those things correctly. And if you leak the information to the model, then it can do much better. So it's similar to like ChatGPT. You can ask it like, write me a book that's going to win the Pulitzer Prize. Okay, the model might be able to do that, but if you go and you give it an outline or you give it like some of the chapters to condition on, you're going to get much better generations. So it's the same kind of thing here. If you prompt with additional information, then you will get like better results.
One more question.
So many biological molecules like proteins and DNA exist in non-canonical forms, whether it's phosphorylation or methylation. How well does Chai do in modeling the effects of those modifications on the resulting structure and binding?
Yeah, so you can provide, we call those are called post-translational modifications and modified amino acids. So you can provide that information to the model as well. I don't know if we have a full write-up of that in the paper, but generally you see pretty good performance. Like it's double-digit % successes in terms of like the model being able to like place those within 2 angstrom RMSD, like where they should be. So, so the model does a decent job with that. One of the interesting things we find is that if you look at previous tools that couldn't recognize that stuff, like AlphaFold 2, the model, so like if you don't provide that information, the model sometimes implicitly figures out that something should be modified, and then it, you might see, it's obviously not going to place the modification because it doesn't know modifications exist, but you might see some like hole, for example, in the structure where like the modification should be. If you go to Chai 1 and the thing should be modified, but you tell the model it's not modified, like you don't put in the modification, you actually get some pretty weird results sometimes, because the way the model is trained is it's like, okay, like if this thing is modified, you should have told me it's modified. And the fact that you don't put in the modification means you're telling me it is explicitly not modified. And that can sometimes cause like weird behavior. So I think this actually illustrates that the model is good at following that stuff, even to the extent where if you don't provide that information, it takes that to mean like it is not modified. So you can think of like not modified is basically like a state. So if we go back to, the examples over here, this, I didn't say anything is modified. Actually, this might be one of the reasons why I got worse performance, because there are some modifications in the ground truth of this thing. So I wouldn't be surprised if I went in and put in all the modifications, I might get a better answer. I think it's actually pretty interesting that the model is like that sensitive to these modifications. That I don't know in this case in particular, because I didn't try it with the modifications, but we have observed cases where we've forgotten to put in modifications and then the results get like a lot worse. And again, that's because the model is actually I think pretty sensitive to those kinds of inputs. So yeah, that's a great question.
So one more about the predicted confidence. Did you test how well calibrated your error estimates are?
Yeah, let me show you that this is in the technical report as well. I lost it. Let me just pull it up again in the browser. Actually, okay, I found it over here. So, yeah, that's summarized here, figure 5. So we're looking at, there's many ways you can kind of dice up those confidence matrices, right? So it's a matrix. What we are looking at for these tasks is something called chain pair IPTM. This is basically taking the average of that matrix where the chains are interacting with one another. And then we're using that, we bin the quality of the results. So DocQ is the metric of how good is this thing? It's basically accuracy. And then we bin it by the chain pair IPTM. And you can see that when the model is more confident, the results tend to be better. And when it's less confident, the results tend to be worse. So we see this for protein-protein interactions, protein nucleic acid. For protein ligand, DocQ is not the right metric. People often are looking at pocket RMSD. Pocket RMSD less than 2 is the setting where people often call this a success. And you find that if your chain pair IPTM is, this is a number between 0 and 1. If it's between 0.9 to 1, you're almost always less than 2, which I think is pretty exciting. So that means that if the model is if the model's not confident, it's possible that it's correct. You still got a bunch of stuff that's good. But I'd encourage you to trust your results. If the confidence is very high, that means you can probably trust the results. And we see in those cases it's basically actionable for drug discovery.
I have a completely orthogonal question. So I'm looking at your GitHub, and it seems like model need to be run locally. And what's the hardware requirements? Is it A100?
Yeah, so hardware requirements, it will depend on how large you're going to do inference. So if you want to do again, an 80 gigabyte A100, you can fold up to 2048 tokens, but you can use other hardware as well. With I know people are using this even on 12 gig GPUs, but they're just having to decrease the constant stop and not doing massive proteins. So yeah, it all depends on what the memory requirements are.
So A100 for inference, and what about fine tuning? Is it enough?
Say again, 40 gigabyte A100?
What about fine tuning?
I guess it depends how you're going to fine tune the model. What's the fine tuning application that you have in mind? What are you going to fine tune it on?
I don't know yet. I need to know what can I do with my hardware requirements.
Yeah, maybe give an example of fine tuning that was successful and the compute that was required to pull it off.
Yeah, I mean, this is what I'm asking. I haven't actually seen anyone fine tune these models successfully yet. I mean, part of this is the model is quite new. When to fine tune the model, I think might be kind of tricky because you'd have to get all the optimizer stuff right and learning rate. There'll be a bunch of experiments there in terms of what you can fit into memory. Will also depend on the optimizer that you use. When we trained this model internally, we even use some custom optimizers in order to help with the memory as well. But at the same time, we actually don't train the model with the full 2048 context size. We're actually usually training. You can train, I think, even if you use 256 context size for training, the model will do pretty well. We go up to I think it was 768 during training, but we didn't train 768 the entire time. Like these models, they actually scale cubically in terms of context size versus how long it takes to do inference. So would highly recommend actually training on smaller crop sizes, even just to get like better performance out of this. So so if you're going to do fine tuning, I think you can probably use like whatever you want. The challenges are going to be on the memory side, like just go with lower crop sizes. Building like the training infrastructure and figuring out like learning rate and so forth. And actually even getting those right crop strategies. Like there's just a lot of details to get right here. And it might require some heavier experimentation if you want to get a really good result. And I think one of the reasons why it's also just like hard to do this is that the baseline performance is already like pretty decent on most of these tasks. So the model is already trained on a lot of the publicly available data we can get our hands on. So some people are like, oh, I want to go and like fine tune this on like antibody antigen interactions. But it's like the model's already been trained on that. So if you fine tune, like maybe it would get a little bit better in that setting, but it probably only gets better if you get all of these details right and spend a lot of time like working on it. I don't think anyone has managed to like fine tune the model and get a better model than like what it can already do out of the box. So I think that's one of the differences here versus like maybe things in NLP, but I think even in NLP these days, right? Like fine tuning the model is, I think even is like quite challenging these days if you want to get better performance than some of the baseline like zero shot performance these days. And I think this is a good problem to have. It just means that like the baseline models are just like getting more powerful over time.
It's got
Understood.
65 billion parameters on my computer.
Got it. So A100 is enough to get embeddings out, and then we can do transfer learning, for example, right?
Yeah, you can try that, for example. Yeah.
Okay. Good.
I also have a model training question. So there are a number of very successful diffusion model tuning methods. Low rank adaptation, or LoRA, is one obvious example. There's also a number of important sort of instruct types of fine tuning, and also reinforcement learning, either from human feedback or DPO, direct preference optimization. And I wonder, in this world of adaptive methods, like what you at Chai see as promising versus like overly challenging. And I guess when it comes to applications, what are the most important legitimate applications of fine tuning? In my mind, obvious examples are you want a particular kind of functionality, which you can describe either with examples or in natural language, and the model preferentially gives you results that look like that, or generates content that is in line with your description of what it is that you want. But I'm curious to know, I guess, what kinds of fine-tuning or model training you see as fruitful. I guess final comment, so the inventions of a number of those have spoken here, and we'd love to put you in touch with them if it would be helpful. That's with Edward Hu, the inventor of LoRA, and Raphael, the inventor of DPO. So maybe there's room for a collaboration.
Well, yeah, I mean, we're definitely very interested in those kinds of techniques. I think, practically, if you think about the users of these tools, we really actually, end game, want this to be biologists, right? And like, biologists are not going to know the difference between LoRA and DPO and RLHF and so forth, right? And I think it's actually these models, if we can get them to work in a zero-shot way, like the dream here would be, can you zero-shot design a drug, put in all the specifications you want, and like get out a drug that just works the first time. That's like the holy grail. And obviously it's going to take a long time to get there. So in the meantime, if we think about like what are the main fine-tuning tasks, it would probably be using the model in the loop. So maybe you use the model to design something, you get some experimental data, and then you feed that back into the model, and then you kind of go again. So those are systems that I'd say like I think we're still early in figuring out the best way to do that. I think all the techniques that you just mentioned, Jeremy, like would be like are the directions to look at. We just haven't gotten there yet. I think there's a lot of exciting work that's going to happen. But the first step that you need, and this is why we started with zero-shot capabilities, is like you need to start somewhere. Like you need some data. You need the model to generate something that is like going to work to some extent before you go and fine tune stuff. It's kind of like doing LoRA on like GPT-1. Like no one really cared about fine tuning back then to some extent because the models like weren't good enough. Once you get into like GPT-3 territory, okay, like now this becomes like really interesting. The models are much heavier, so like LoRa versus like full fine tuning becomes important. So maybe what I'm saying here is I actually don't even know the answer to the question about like what's going to be most useful or how it's going to be used. I think that there's still some more work to go. I think if you ask us in a year from now, I think we'll have like a much better understanding. So definitely some stuff that we plan to explore in the coming months. But it's a yeah, very, very exciting direction for sure.
Hey Joshua, a few questions I have. One is like related to ions. Like, do you particularly deal with like ion binding because they may cause some conformational changes? This is the first question. The second question is about like dynamics. Like, does the model have a capability to model the dynamics of the protein and ligands, etc. The third one is related to like when you have binding, like how, I mean, have you ever checked the binding affinity, like whether the model can predict the binding affinity like accurately, or extend as like something like the IC50, like efficacy, etc. in real world like application about like drug discovery, because like they are kind of the direct downstream before you actually jump into the active learning loop for drug design. So I wonder whether those properties has been monitored and whether the baseline model can capture those correctly already, or there is still some room to improve in the future.
Yeah, those are great questions. I would love to know the answer to most of those too. Reality is I don't know. That's one of the reasons why we open source the model actually. The evaluation is actually a lot of work, right? To go and like answer those questions very thoughtfully. And one of the reasons why we put this out is we hope that the community will help us to answer those questions about what the models are capable of. I think something like dynamics, people often think, okay, like there's a diffusion model here, so maybe we would like sample kind of like a conformational diversity. My intuition is that it won't work very well for that. I think where the model might be able to capture some of the dynamics is more like, here's a protein, and then here's a protein when I put it in the small molecule, and then have the model like maybe reform the protein around that small molecule. Like I think you might see some of that stuff, but this is just my intuition. Like we haven't actually explored this kind of stuff yet. So something I would love for someone to explore pretty deeply and tell us. And especially do it in a quantitative way, right? Like I'm kind of giving that intuition from playing around with some examples and just like my understanding of like what I think the model should be able to do, but definitely lots more that we need to explore here.
Okay, thank you. So a question about the research progress in AI agents as it applies here. So early in agents, there were papers like React, which described specific evaluation loops that agentic systems could use in order to do things that were much more complex than the base model can do. When it comes to zero-shot drug discovery, what is the set of tasks inside the agentic loop that enables zero-shot drug discovery? And if you were to sort of say in 3 years it's solved, what is like the high-level design of that agentic system? What evaluation processes is it using? What is the base model's application sets? And yeah, I guess try to make it succinct, but if we were to like say propose the base architecture and treat it as a research problem fulfilling every part of that agentic architecture, what would be the parts of it?
Yeah, it's an interesting question too. So I think probably how you would kind of set up an agent, how you would kind of frame this in an agentic setting. So if you look at Chai-1, you know, it can predict the structure of a protein in a small molecule, right? For example. Usually a drug designer has a certain specification they're trying to reach, right? They're looking for a small molecule that has a certain affinity to a target. It's also not binding some other target. Many of these questions can be answered with the model, but you need to enumerate them, and you need to make changes to your molecule until those features are kind of explored. So you can imagine a situation where the model, you start with the model, like the agent starts with some like base small molecule, and then it says, you know, this thing looks like it binds like only somewhat well. Let me make some mutation to that small molecule. Let me make some modification to that small molecule so that it fits better into the pocket. It makes that change, it refolds, and it's like, okay, this looks a little bit better, but now I need to go change this part in order to fit in better. And basically it can go through an iterative loop where it's like making modifications to the small molecule. Ideally, those are modifications that can actually be reached with chemistry. Like not every molecule is actually easy or achievable to make in the lab, but the model might, the agent might actually know what, and you can train it to like tell it like what are the actual kind of reactions that are feasible for chemists to do. It can go through that whole process, and then it can actually come back with like a recipe of like, here are the things that you should go make. And perhaps we're in multiple trajectories. Like I think what we might, if we imagine drug design 5 years from now, it might actually look like agents doing a lot of the intellectual exercise over here, and then going back to chemists and saying like, oh, here are like the steps in the process to go make these molecules. I think that I have high confidence that among these 30, a couple of these should actually fit your specification. So go and make these, and here are the instructions. And maybe that's like 5 years from now, maybe like 5 years later, it just becomes even a whole robot that then goes and actually experiments on the whole thing, and we just like cut humans out of the loop entirely. I actually think that the AI part of this will happen faster than, let's call it, the mechanical part of actually doing the experiments. It's kind of crazy, but I actually think solving drug design on the computer, like in terms of like what, like how to design a molecule, will be easier than like actually having robots that go and like do all the lab work. And I think we're seeing similar things in the rest of AI as well. Like self-driving cars might be harder, or seem to be harder than just like language models that can like do legal work, for example, in terms of like getting all the edge cases down. But I think these are all, in the future, I guess like all of us are going to be cut out from the process. Like AI, I guess, should be able to do a lot of this. I think AI is uniquely suited for drug design, just because like you and I, like all of us in this room, have like very, very little intuition of like how a DNA sequence will transform into some 3D structure at the end of the day. And it turns out that there are chemists that have built those intuitions over decades, but it doesn't come close to like what a neural network is able to do when it's trained on all of this data. So I think as the methods become more powerful, it's a very promising time, and we should be trusting I think the methods more than humans. And the next barrier that we have to hit is like being able to trust the computer more than like random search. Like most of drug discovery is called discovery because we just try a bunch of random things, and there's a lot of like really good processes that exist for testing these things at scale. And that's with like the virtual screening and design and agents and all this stuff, I think needs to get a lot better. So we're still, I think, in kind of like the GPT-2, GPT-3 kind of moment in this space, where these models are showing signs of life. They're very useful in a couple of settings, but we really need to push them farther in order to just like completely replace the methods that people are using today in the lab.
Les, give us 3 specific examples of Chai contributions that are in the shape of hackathon projects that would be genuinely valuable to the open research frontier, but that are doable today.
Yeah, these are going to be more like on the biochemistry side. There's let me give 3 of my favorites. So there's a group in the UK that's been using Chai in order to create a variant of a molecule that's FDA approved, but like 40% of patients get resistant to it. So they've been working on this project 3-4 years. They don't know what the 3D structure looks like. It's been very hard for them to like get the 3D structure in the lab, but they typed in the molecule they're working on into Chai-1, and the scientist, like, I fell off my chair. Like, I finally understand like how this molecule is interacting with the protein, and now they use that to go launch another battery of experiments. They've been basically what I described the agent of doing. They have like they have like scientists doing that, and they have been iterating through different molecules and now getting promising lab results. So that's area number 1. I think is really cool. Area number 2 is people are using Chai-1 for screening different enzymes. So enzymes are proteins that are, you can imagine them like interacting with small molecules and catalyzing them. So they can screen through many designs with Chai. So you just like, brute force all these designs and check where does the model have high confidence, and they use that to filter down what they're testing in the lab. So it's really accelerating enzyme design in that way. And then maybe the last example I really like is like for antibody antigen design. So again, it's still hard to design antibodies on the computer, but if you run many samples from the diffusion model, you can start to get you can sort of like boost performance by even an order of magnitude. So people are doing that, trying to get predictions of like how an candidate antibody drug fits in with an antigen, and then use that to kind of ideate what are some rational changes that you can make to get that antibody to bind more tightly. So I love these three examples because they're just like all so different from one another, and I think really highlight how these general purpose models can kind of be used across the industry. But we're still very much in the early days of this stuff, and I expect we'll figure out much more, especially as the tools develop. Again, Chai-1 is really just a tool. It's not yet a product, and it's also, you know, I think not the final stop of this technology. I think it's really the beginning of a story rather than an end. So I think there's a lot of opportunity to make these things even more useful for scientists.
Thank you so much. Let's have a huge round of applause for Chai Discoveries, Joshua Meyer, and Paul Ronhard. Thank you. Thanks, everyone. All right. And if you end up winning the hackathon, get the opportunity to have lunch with the wonderful folk at Chai Discovery. So, little incentive for you all. Thank you.
Well, thank you.
Okay. So this is the moment where we turn the mic over to you, the participants, and discuss the projects that are on offer today at Hackathon. Now, there's a spreadsheet that has been sent out, basically listing the likely projects from folk at Hackathon. If you're on this list, that is fantastic. But if you're not, we're going to spend the next 2 minutes. Take whatever idea you're currently thinking of, maybe it's fine-tuning over Chai, and just list the likely project in this spreadsheet. And I guess I'll request silence so that anyone who hasn't put it in yet has a chance to do it. So 2 minutes, it's sent out over both Partiful and Slack. So please, everyone, add your likely project to the spreadsheet, please, next 2 minutes. Silence while everyone's working. You guys should wait to chat until we're done. Thanks a lot. $30 seconds left. If you've not completed your proposal, then please do. So time to step into our lightning talks. The idea is pretty simple. It's just a 1-minute pitch of your project that the folk have. And so I'll just take it from the top. Yeah, if folk are here, raise your hand. Most of these folk are here, so keep it. Lana and Kyle, are you here? Interested in discussing chronic disease management? I don't think Jason's around, but Justin, would you like to give a brief lightning talk on understanding thyroid? Justin? Oh, see that? Wow. Oh, see, sorry, all of these were like filming.
Yeah, before.
Okay. Okay, so I'll open with like a lightning talk. So like, I am very interested in research automation, and we had a breakthrough on machine learning algorithm generation, where it turns out that if you come up with the right domain-specific language, in our case, specifically like a scikit-learn style domain-specific language, where your generated algorithm comes with a fit and predict function that you can automatically evaluate the generated machine learning algorithm on a suite of downstream benchmarks. And so by that process, you can just use prompts to search through the space of machine learning algorithms. And if you have a really creative meta prompt, you can generate hundreds or even thousands of downstream descriptions of algorithms that are nominal descriptions, have a code generation model implement them, and then do automatic evaluation on 12, for example, real benchmarks to see which ones work. And so I want to create this automated eval pipeline in the context of biomedical research. And the idea is really to find algorithms that are relatively simple, such that a single prompt can describe them, that can be automatically evaluated in a really concrete way, where the evaluation can't be fake. So that even in the face of generating, for example, like hundreds of thousands of hypothetical algorithms, you trust the evaluation process to accurately filter out the ones that are outperforming. So yeah, if you're interested in automated eval pipelines for algorithmic research, do let me know. Dan, in here, let's discuss LLM and synthetic lab purpose. 1 minute.
Sure. So it's kind of in line with what Jeremy just talking about, the synthetic biology protocols, right? After you have the idea, after you have this molecule, you need to execute it somehow. And this is where protocols comes in, that for people who don't know, protocols is basically like recipe for cooking, right? What do you need to add together, what do you need to stir, what machine do you need to use, and so forth, to get certain result. There is a goal and there is a result. There is a data set available, you know. You can ask right now ChatGPT to create a protocol, and it will produce something for you, but you need to evaluate or validate it somehow, right? How do we validate? Obviously, ground truth is, you know, lab in a loop, but what can we do in silico? Okay? How can we validate in silico these protocols? And this is the problem that I would like to solve today. We might use Chai because plasmid sequences are available. We might do some, you know, unsupervised learning for clusterization, you know, search and so forth. But that may be too much for today.
Thank you. Okay. Okay. Question. Would you like to discuss value prognostic model?
Hi, I'm Gretchen Borgen. This is Jorge Jimenez. We are envisioning a cloud-based, cloud-guided tool that would help predict the longevity reductions due to typically predictive disease, such as heart disease and diabetes. So we're hoping to combine a predictive model for heart disease and for diabetes with EHR and health kit data, wearable data, to analyze a complete picture of what your longevity might look like. And in addition, bring attention to potential comorbidities and diagnoses that you may also have to, by association, monitor.
Real quick on the approach, we're going to do some basic stats, you know, try to discover the data and then eventually use Claude as well. Sorry.
He'll elaborate later.
Thank you. Pablo, would you like to discuss correlating PLM embeddings with chemistry centers? Okay, Grace, Arifan on RNA therapeutics.
Sure. Rubicon is not here. He is in Massachusetts, but I'm going to use one of the AI tools that he created to predict the molecule the RNA will react in the body to data systems. So there's a lot of different RNAs that can be used to intersect at certain pathways that are associated with inflammation to turn off or on the proteins in the pathway. And there's just so many combinations of the RNAs, and we can use some type of AI to create all of the RNAs very quickly and test them. This isn't necessarily AI, but you can use Chai to implement them with the structure of the enzymes as well, and check if they do fit together. So that's another way to implement the AI. And then using the
next one to test the immune response in the body. It's basically predicting the body's response and the cell's response at the same time with different kinds.
So yeah, I guess I listed another idea. So the thought is pretty simple. A lot of machine learning models will come with fine-tuning frameworks, and the idea is just to find some very simple examples of data with which we can do a LoRa and DPO fine-tune of Chai-1 and create a GitHub repo where anyone can download it, and if they have a lot more data than we do or a clear idea of exactly what kind of data type they want to fine-tune for, they can drop it into a training pipeline that we've set up. And we would provide an example of applying these techniques to Chai-1. Plausibly, we'll also ask the Chai team to give some guidance on exactly what kind of training setup they did so that when we replicate it, we don't make any egregious mistakes. But I do think this might be the first, since it's the first sort of big open source variant of AlphaFold that as far as I can tell is fine-tunable, it might be original research that ends up being publishable, and I'm happy to invite Robin to sort of collaborate on if it turns into a functional repo that the community can use to train custom models. And then I think the high-level motivation is, I do think controllability will become a big issue. So Josh, when he was describing the agentic system that creates a drug from scratch, would say, oh, well, actually, this didn't bind quite well enough. Let me improve it in some specific way. And that kind of guidance usually requires instruct fine-tuning. And so the idea to use language the way that he was to try to guide these models is something which could be an add-on that could be quite powerful. So that was one of the thoughts. Yeah, maybe there's another project which is like a language add-on that lets you control the generations using natural language. AJ Nando.
This is 15 seconds, super weak. I'm just trying to get my head around using multiple agents to present multiple perspectives about clinical, patient, physician, and treatment strategy. Just get my head around this.
Robin. Yeah, so I was watching Joshua's presentation. I was really inspired. I'm inspired by when stuff goes wrong, and so he gave us the example of where the model got a low confidence score on the docking, but it had pretty decent matching on the individual components, and so there's a bunch of ways of inputting the data that we saw on the GitHub. You can do ligands, you can do the full protein chains, you can do structural templates. So I just want to make a pipeline that tries all of them and then sees if that increases the confidence score of the model that's trying to do.
Great. Any other intrepid product descriptions? Going once, going twice. Okay. Yeah, so I mean, it looks like at least if people are successful, and at the end of the day, we make a lot of progress. So I'm quite excited. So yeah, I guess a few final comments. As you take the next 20, 30 minutes to hang out with each other, discuss really creative ideas that have come up, and decide what project to do. We'll check in about half an hour to make sure that everyone is sort of settled and what they're up to. We've created a ton of time, and I'm really happy about that because we do actually have a really solid 9 hours in front of us where we can knock out a project that is as big as, for example, a fine-tuning project or figuring out how to do protocol generation successfully. So yeah, do take it as a glorious opportunity. I will say that there are just so many examples of being at the frontier here today, and in many ways, sort of we heard from Josh, there are so many projects that have just never been tried. And so it actually is a fantastic moment to be doing research because the low-hanging fruit is pickable and you can very quickly become a part of the history of these models. And I will say that a lot of these tools they exist, but the level of engagement from the machine learning community is so low that coming with a little bit of ML knowledge can be incredibly powerful. coming with a little bit of infrastructural skill can also be incredibly powerful. And forming teams with folk who have a totally different form of expertise often can turn you two into the only team working on a problem of interest with that kind of expertise. And so, yeah, it's just really a verdant opportunity. Any like last minute questions about what we're up to? Oh, sure. So prizes. So Anthropic has sort of wonderfully sponsored this event with their credits, but there's also going to be $1,000 of Anthropic credits given to the top 3 teams from the hackathon. Secondly, there's a Chai Prize, which is you'll get to go to their office and meet them, and they really are fantastic. They put out an alpha flow to your model. There's so much to learn from, and so many really potentially impactful applications inside that. So
You'll double your credit on Chai as well if you win.
oh, sure. Oh, cool. Great.
They'll do what then?
They'll double your prediction limits on Chai.
Oh, okay.
It's another form of credit.
Okay, and then I guess some logistics. So currently you're on the 3rd floor of a 4-story mansion. There's 2 floors below this one. Please do fully explore, find a nice spot. The food for lunch will come in about an hour and a half, and dinner will be here at around 5. There's a bathroom in this left-hand corner, and there's another bathroom all the way downstairs on the base floor. Please do not go upstairs. Those are private bedrooms, so we're going to try to keep it isolated. Yeah, I guess there's a lot of potential in these projects, but I do feel like it's surprising to me that the medical science and longevity angle of attack using artificial intelligence has been so undersold. And so part of my hope for this event is we can ship projects which can galvanize a field that will realize, that will wake up and recognize that it's possible to make a ton of progress in bio by using techniques like these. And in my mind, the plausibility of success of reaching the longevity bridge via AI and technological progress that's software-centric is so prominent and really so filled with hope. And really this can be, in my mind, the central purpose of a lot of people who probably don't realize that this technology is burgeoning and has incredible capabilities. And yeah, the fact that open source is here is really a gift. There's a real sense that bio could have been just as closed as it was, and machine learning culture could never have sort of taken over. But all this stuff is on LiveArchive or open on GitHub, and you have the opportunity to interact with every single aspect of the model. You can run it yourself on your own hardware. It's not going to be taken away from you. Companies aren't going to hold back the capabilities frontier for their own sort of private sort of financial incentives. And currently, open source state of the art is exactly aligned with private state of the art. So actually, it's an incredible opportunity moment to execute on it in these next 9 hours. Lots of love. Looking forward to seeing projects that you deliver. Yes.
Can you provide the Wi-Fi password?
Yes. So the password is attention is all you need. Wi-Fi is AGI House, the paper you may know and love.
So the Anthropic credit can come? Sure.
If you didn't sign up in time, sorry.
I signed up in time before.
It didn't come?
Yeah, it didn't come. I'm not sure about that.
Oh, okay. Yeah.
Troubleshoot later.
We'll follow up with them and make sure that it's coming.
And how can we use Anthropic today? We can use time model in Anthropic or?
So Anthropic has an image API and an LLM API. Yeah. So if you're doing anything with, say, biological image data, like Spectre, for example, or something like that,
we can fine tune on it.
Agentic loops. I don't believe they have a fine tuning.
Okay.
But yeah, I'm pretty sure OpenAI came out with one if you want to try that. And then I guess for context, Anthropic also sponsored our physics hackathon, and in that case, they came through a credit for halfway through the credit. So they'll come through eventually. Just the whole thing. Any other questions? Thanks for the question. All right, very cool. And with that, let the AI for medical science and longevity hackathon begin.
I have a quick question about the documentary stuff. I think that could actually be a really great submission to try. Would be here's how to auto detect the residues in the context of the specific subsets of the sequences that are actually associated.
That would be useful. It seems like you were saying that it would be
Hey, who was it who talked about protocol generation?
It was the guy with the ponytail and her.
That's actually much more interesting. Are you going to be pushing forward with that?